You guys can say hi. Hi guys. Hey, so this is the surprise demo that we were talking to you about before. And we're all gonna do this for you, and we're all gonna do it together. If it was not pandemic COVID-19 year, we would get to do this with you guys, so we're sorry about that, but we still really wanted to make sure that you got a chance to see this happen. So we are answering the question today, what does DNA look like? And what we're gonna do is we have 3 different fruits right here, if you can see them, which you probably can't, go like that. We have a strawberry, a banana, and a kiwi, and we are going to try and remove the DNA from these fruits, and you're gonna get to watch us as we do that. So in terms of safety, you can see we have our safety glasses on. We're wearing lab coats. Normally we would just have you guys make sure that you're not wearing like your favorite shirt or something like that, so that we could be careful with everything that's happening. We have a lot of different materials here. We have some salt that we're gonna be using and some dish detergent. We have hot water that's getting ready at the sink. We have filters and funnels and test tubes and test tube racks. And of course, we have a set of procedures and directions that we're gonna follow. We're going to properly demonstrate how to follow directions, because my kids know I make a big deal out of that all the time, and I'm sure they do too. All right, so the first thing we're gonna do is we're each gonna get 50 milliliters of hot water, and we're gonna put them in our Ziploc bags. So we have our Ziploc baggies here. I'm going to be working on extracting the DNA from a kiwi. Mr. Krebs is going to extract the DNA from a banana, and Mrs. Mistrella is going to get the DNA from a strawberry. So not boiling water, but hot water, and we're going to put that first in a graduated cylinder. That's what we use for measuring. After we get the 50 milliliters of hot water, we're gonna add a teaspoon of salt. Now, a teaspoon of salt is kind of like a nickel-sized portion in your palm. Yes? Yeah. Salt. Okay, pass that on to Mr. Krebs. So I'm going to just kind of mix the water. We're gonna not violently at all. The bag isn't even closed, and we're just gonna try and get the salt to dissolve. So when you stop seeing salt crystals, salt chunks, stuff like that, you want to try and get most of it dissolved. You'll still have some. We'll still each have some granular salt in there, but we do want to get kind of like a salt water solution. It totally wouldn't taste good. Kind of like the ocean. It's never fun. All right. So after we do that, after it gets dissolved, we will each take our chunk of fruit. So here is my kiwi chunk, and we'll put the kiwi chunk in the bag. And now we are going to mix the fruit with the saltwater solution. So we will close the bags carefully this time. When we do this, basically, we're kind of working on making a fruit smoothie. That's what we're doing. Oh, I should have gotten a more ripe kiwi. We're kind of trying to make a fruit smoothie, all right? Some people in the scientific world would call it a slurry, actually. If you watch enough NCIS or Bones, you hear that a lot. So we're making Criminal Minds. We're making a slurry, and it's gonna change colors, and it's kind of gonna feel squishy and gross. So another reason why we would love to do this with you guys, because this part's kind of fun. Is this part fun?
This is fun.
Yeah. Are they squishing? Are they squishing nicely? Okay. My kiwi might have been too ripe. I'm not sure. So we're gonna continue to get this all mixed together, and we're probably gonna spend about a minute making this slurry. Notice we're being gentle, we're not going crazy. All right, we're getting, they're all changing the right color. Oh look, we did it colorfully too. Red, bananas are white, yellow, yellow, green, kiwi, okay. Now what we're going to do is, once we feel as though we have a good slurry there, a good mixture, we're going to add a small squirt of the dishwashing soap that we have. And we're aiming for about the size of a dime here. I don't know if we want to do it this way. We might want to even just use the squirt bottle.
Got it.
Yeah, we're just going to use the squirt bottle.
You can squirt.
Yep, because look at that. That's a lot of soap. All right, we're going to just put a squirt bottle in. There we go. About a dime portion. Now, here's an important step, and this is one that isn't often followed. All right, so I'm going to say it nice and clearly so that my colleagues over here follow the direction. Okay, the direction is to gently mix so you avoid bubbles, not shake, okay? Not shake, we're just mixing. We want it to mix, we need it to mix, okay? So while we're mixing this for just a little bit, I kind of want to talk about why we used what we used, okay? So these are all fruits. Fruits are all plants. Plant cells, we know, have a couple different organelles than animal cells. They have a chloroplast that's different, and they have a cell wall that's different, and they have a very large central vacuole. We're going to focus on the cell wall one right now because DNA is in the nucleus, and the nucleus is in the cell, and the cell wall is on the outside. So first, we got to get through the cell wall. And that's actually what we use the hot water for. We use the hot water so that we can basically break down the cell wall of the plant cell. So the cell wall is gone, but now we have a nuclear membrane that we need to get through. And we have a cell membrane. So we have two membranes. And remember, those membranes are made up of phospholipids, which are fatty acids. So we got through the cell wall. Now we have to get through the cell membrane, and then we have to get through the nuclear membrane. Well, that's what the soap is for, the dish detergent is for. That's going to actually break down the fatty acids of the cell membranes and basically expose the DNA for us. So in here right now, in each of our bags, there is DNA. We have broken down the cell wall, we have broken down the cell membrane, and there is DNA. Now we have to go to another step that's really important, which is we have to make it so that we can see the DNA. So we have to separate the DNA from everything else that's in here. Okay, so what we're going to do with this step is we have our coffee filter and our funnel and our test tube, and we are going to carefully pour the contents of the Ziploc bag into the test tube, into the funnel, and we're going to keep an eye on the test tube so that we don't overfill it. Okay, and this can get messy, so we're going to be very careful to do this very nicely. Oh boy. Here we go. Oh, there we go. And then the chunks. Okay, all right. So mine is in. I'm gonna grab the trash bag for all of us. Trash can for all of us. Right behind you, guys. And you can see, if you watch, each of ours is going to go through. And we're gonna have to let this sit for a little bit. Now, we're going to make sure that we can get as much of this out as possible. So we might squish it with our hands very carefully so that we can get all of the fruit smoothie out, we'll call it that, all the fruit smoothie out. And then what we're going to do is our next step is going to involve cold isopropyl alcohol. No, yes, yes, cold isopropyl alcohol. Isopropyl alcohol is rubbing alcohol. Ms. Mistrella, do you want to grab the isopropyl alcohol? We're going to use cold isopropyl alcohol, and we're going to add that in next. Now, the isopropyl alcohol, we talked about how there's DNA in this right here. Okay. Did you want to tell them about isopropyl alcohol? No? No. Okay. I can talk forever. My kids know that. We're having fun here. All right. So, the isopropyl alcohol. Right now, we have green juice, red juice, and bluish juice because the soap is darker than the banana. All right, and the DNA is there, but somehow we have to separate it so that we can really see it clearly. And what's going to happen is the isopropyl alcohol is basically going to allow for the DNA to coagulate and kind of separate itself from the rest of the contents. So we put the salt in earlier because the salt is going to make all the DNA that we took out, it's going to make the salt stick together, okay? Or I'm sorry, it's going to make the DNA stick together. And now what we're going to do is we're going to separate the DNA from all the other stuff that's in there, and that's what we're going to use the isopropyl alcohol for. So I'm going to just kind of try and squeeze a little bit more out of mine. We don't want to get chunks in it. That's one of the problems. We don't want to get chunks through the, there we go. We don't want to get chunks in the test tube. That is not helpful. But we do want to make sure we get a decent amount of liquid into each of them. So we're kind of just moving some through. I'm actually just pulling the chunks of kiwi out of the way of the rest of it. Okay, and then mine's ready, so I'm going to throw out my coffee filter. Carefully, I'm gonna throw out my coffee filter. And put my funnel here. And then I'm gonna start out by putting my isopropyl alcohol in. Now the isopropyl alcohol, it is kept cold. It's kept in the freezer, all right? And so here it is, right here, you can see it. And what we do when we put the isopropyl alcohol in order to be careful with it, the proper way to add materials to a test tube is to turn the test tube kind of on its side, and then to let whatever substance you're adding in, basically to squirt it down the side so that it doesn't just all get kind of crushed in there together. Because remember, we want to separate the isopropyl alcohol from the rest of the substance. So if you look at my test tube carefully, you can see there's definite green and then there's definite clear isopropyl alcohol. So we're each going to do this, and then after we do that, we're gonna come back, we're gonna let it sit for 10 minutes, then we're gonna come back, you'll watch part 2 of the video, and we're gonna see if we can actually get some DNA to show up between these 2 layers of isopropyl alcohol and the fruit smoothie that we have made. Alrighty, we will see you guys back in a couple minutes.